Good morning, students. Whether my voice is audible?
Yes, ma'am.
Just a minute.
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
Yes. Whether my voice is audible?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Fine. Today we are going to discuss regarding the concept of molecular basis of inheritance. We are going to continue the concept. Okay. Then let me present the presentation. Let us brush up certain concepts. I hope screen is visible to everyone.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, just a minute. Fine. So yesterday we discussed regarding the deoxyribonucleic acid, ribonucleic acids. RNA, DNA. Then after that, the structures, nitrogenous bases, purines, pyrimidines. Right. So today we are going to discuss the other concepts like the experiments, complementary strand, leading strand, and lagging strand. These all questions we are going to these all questions we are going to discuss today. Right. So here in the DNA, during the complementary strand, we have to remember one concept regarding anti-parallel, parallel and anti-parallel strand, 5' end and 3' end. And remember, the complementary strand twisted and the distance will be 34 angstrom. Each twist of DNA, each twist will be containing 10 base pairs. And the distance between these two base pairs is 3.4. That we have to remember. Okay. Next. DNA structure in okay. Fine. So packaging of DNA in prokaryotes is also a very important concept. 2 or 3 questions. I mean, we are going to discuss 2 questions on it today. I request. Okay. Saitra, can you read out the slide regarding packaging of DNA in prokaryotes?
Yes, ma'am. Prokaryotes do not have definite nucleus. The DNA is not scattered throughout the cell. It is held together with some proteins in a region called nucleoid. The DNA in nucleoid is organized in large loops, AB proteins.
Okay, held by proteins it is. Fine. Thank you. Thank you, Chaitra. Kanchana Raj, can you read out packaging of DNA in eukaryotes?
Yes, ma'am. In eukaryotes, DNA is stabilized with positively charged basic protein called histones. Histones are positively charged due to the rich basic amino acid like lysozyme and arginine. Histones are organized to form a unit of 8 molecule called histone octamer.
Okay, histone octamer. Here, I know you guys read it in theory, like in theoretical exams, you read it. Now I am going to repeat it because while answering, it will you will be confident to answer because some of you just answer by copying the other messages. And see the diagram properly, guys. DNA, that is histone 8 proteins. See this nucleosome structure properly. Okay, then one more slide, then we will repeat with the we will go to the questions. Negatively charged DNA. DNA will be negatively charged, wrapped by positively charged histone. So together it forms a structure which is called nucleosome. That we have to remember. Then nucleosomes hai kaantave. The nucleosomes are seen on a beads on a string. As you can see in this diagram, beads on a string, structure under electron microscope. Nucleosome forms the repeating unit of a string of nucleus called chromatin. So it forms a string which is termed as chromatin. Now let us move to the discussion of questions. Let us begin with the first question of today. Just a minute. Yeah. Okay. I hope screen is visible to everyone. Yes?
Yes, ma'am. Yes, ma'am.
Okay. So we discussed few of important questions yesterday. Today we are going to continue that. First question of today, which is the correct complementary strand of AGAATTCGC? Options are given below. They are complementary strand. Until I complete the question, please don't answer through mic. Okay. So option A, CTCCGGATA. Option B, GAGGCCTAT. Option C, TCTTAAGCG. Option D, GTGGCCATA.
Option C, ma'am.
Okay. Kanchana, Lakshmi, Anusidi, Veeresh, Deepti, Megha, Chavan, Gayatri, Prema, Ramya, Saundarya, Pavitra, Pallavi, Deepti, Vidya. Okay. Shweta ji. Most of them voted for option C. Kavita, option C. What about YouTube students? Likita, option C. I think Likita was absent for a few days.
Yes,
Fine.
ma'am.
Okay. Fine. So let us see which is the correct answer. I got few more answers from Akshay Patil, Madhumita. Good. Be interactive. So option C is the right answer. Complementary strand. It is a very easy question. A always quotes for T. G, C, A, T, T. Like this, we have to write the complementary strand. Good. 24th question goes like this, guys. Which of the following enzymes are used to transcript a portion of the DNA into mRNA? Option A, RNA polymerase. Option B, DNA polymerase. Option C, protein polymerase. Option D, hydrolase.
Option A,
Option
ma'am.
Option A, ma'am.
Vinayak MK. I appreciate the way of answering and Likhita. Okay.
The name
And
of RNA polymerase.
I'm requesting from the first class, please do answer once I complete reading the question. Until I read last option and then say you to answer, until that, please don't answer. Okay. Because ನಿಮಗೆ ಆನ್ಸರ್ ಗೊತ್ತಿದ್ರೆ I appreciate it. Most of them will not be knowing the answer. ನಿಮ್ಮ ಆನ್ಸರ್ ನೋಡಿ ಅವ್ರು ಆನ್ಸರ್ ಮಾಡ್ತಾ ಹೋಗ್ತಾರೆ. Okay, thank you. So most of them voted for option A, RNA polymerase. 2 or 3 of them voted for option C, protein polymerase. Let us see what is the answer. RNA polymerase is the answer. Here transcription is carried out by an enzyme which is called RNA polymerase and number of accessory proteins. RNA polymerase ashte alla, adar joti accessory proteins irtave. Those are called as transcription factors. Okay. 24th question. Which of the following occurs in the process of central dogma reverse? Central dogma reverse ಅಲ್ಲಿ ಯಾವ process occur ಆಗತ್ತೆ ಅಂತ ಕೇಳಿದ. Option A. How many times I said you? Can't you understand? Don't you understand? Until I complete the question, please don't answer in the chat box. Otherwise, I will just block you all. It disturbs other students. Please do understand. Conversion of a strand of mRNA to a DNA, changes in the chromosomal karyotype, gene replication process. Option D, protein production process. Which of the following occurs in the process of central dogma reverse?
Option A.
Option A, ma'am.
Vidya, Kavita,
Option A, ma'am.
Manish.
Option A, ma'am.
Most of them answered through mic. And option A. Okay, Shamita, Shanti, Bhumika, Megha Chavan. Option A, that is conversion of strand of mRNA to DNA. 24th, A is a correct answer. I request Gayatri to read out the slide. Can you read it, Gayatri?
Yes, ma'am. In some viruses, the flow of information is in reverse direction, that is, from RNA to DNA. This process is known as reverse central dogma or reverse transcription. It takes place in viruses, cellular organisms, where RNA replicates via a DNA intermediate. A complementary DNA molecule is formed from genomic RNA with the help of an enzyme called reverse transcriptase. Example, HIV, which is a retrovirus.
So example of it is HIV, which is a retrovirus, where you can see reverse central dogma. Good. 25th question. What is the name of this nitrogenous base? Uracil, thymine, cytosine, guanine. Deepti, option B. Yes, Harshita.
Option B, ma'am.
Veeresh, option B. Pavitra, option B. Okay, fine. Most of you are voting for option B, that is thymine. Fine. So, illi just remember the structure. Double bond O ide, NH, double bond O, NH. Okay. Most of you voted for option C, thymine. Okay, let us see what is the correct answer. Let us discuss about it using the PPT. Then you can only answer me which is correct. ಆಮೇಲೆ ಹೇಳ್ತೀನಿ ಯಾವ್ದು ಕರೆಕ್ಟ್ ಆನ್ಸರ್.
Hello.
Okay.
Yeah, this is Ravana here. My ID REVANNE.
Yes.
Voice not
ID REVANNE.
Yes. Who is this, please? Someone is asking something, but I am not able to hear. The answer is option A, uracil. It is not thymine. In thymine, again, this bond will be linked to another functional group. But in uracil, this bond will be free. So answer is option A, that is uracil. Just a minute. Okay. 10. Which of the following ensures the stability of the helical structure of a DNA? Option A, presence of repetitive structures of a DNA code. Option B, stacking of one base pair over the other. Option C, presence of aneuploidy. Option D, occurrence of chromosomal rearrangement. ಏನ್ ಕೇಳಿದ್ದಾರೆ ಅವರು? Which ensures stability of helical structure ಅಂತ ಕೇಳಿದ್ದಾರೆ. So what is the answer? Sanvi, Vinayak, Asha, Veeresh,
Option
Parvati,
A.
Saurabh. Okay. Okay,
Option
most
A,
of
ma'am.
them answering as option A. Few of them voted for option B. Darshan, option D. Okay. What about you, YouTube students?
Option A, ma'am.
Sweta. Question not understanding. ಕೆಳಗಡೆ ಒಂದಿಷ್ಟು ಪಾಯಿಂಟ್ಸ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ಆ ಪಾಯಿಂಟ್ಸ್ ಅಲ್ಲಿ ಯಾವ ಪಾಯಿಂಟ್ ಸ್ಟೆಬಿಲಿಟಿ ಆಫ್ ಹೆಲಿಕಲ್ ಸ್ಟ್ರಕ್ಚರ್ ಅಂದ್ರೆ ಸ್ಟೆಬಿಲಿಟಿ ಆಫ್ DNA ಗೆ ಸಪೋರ್ಟ್ ಮಾಡತ್ತೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. So most of them voted for option A. Few of them voted for option B. Let us check who is correct. Option B is correct. So you can read on a slide. Two factors are mainly responsible for the stability. One is base pairing between complementary strands. The other is stacking between adjacent bases. So here, the presence of repetitive structures anta ide. That is not related. The right answer is stacking of one base pair over the other. So option B is the right answer. Let us brush certain concepts now. Okay. Just a minute. Whether the screen is visible?
Yes, ma'am.
Okay, fine. Now let us discuss a few points regarding a transforming principle. Ravi Kumar N. Can you read out the slide, please? Yes, Darshan, any doubt? Okay, fine. Yes, Harshita, can you read out the slide?
Conducted an experiment on mice and pneumonia bacteria, Streptococcus pneumonia. This bacteria are found in two strains, has one virulent smooth strain, seven non-virulent rough strain. The F strain bacteria produce capsule and pathogenic. The R strain lacks capsule and non-pathogenic.
Okay, thank you, Harshita. S strain and R strain we know. Just visualize this picture. When rough strain is given, mouse will be alive. Smooth strain is given, mouse dies. Heat killed smooth strain, mouse is alive. Rough strain mixed with heat killed smooth strain, mouse dies. Okay, this is regarding the Friedrich Griffith experiment. Now, just a minute. Yes, Akshay Patil, can you read out the slide, please? You said no in the message box, right? Regarding something I did not understand for what you messaged no. So can you read out the slide? Is it possible for you?
Yes, ma'am.
Yeah, please. Akshay Patil, can you read? ಯಾಕೋ ತುಂಬಾ ಜನಕ್ಕೆ ಓದಕ್ಕೆ ಬರಲ್ಲಪ್ಪ ಇಲ್ಲಿ. ದೀಪ್ತಿ, ಕ್ಯಾನ್ ಯು ರೀಡ್ ಔಟ್ ದ ಸ್ಲೈಡ್?
When the R strains are injected into the mouse, it's non-pathogenic and does not cause pneumonia. The mouse continued to live. When the S strains are injected into the mouse, that causes pneumonia and mouse dies. When heat kills S strains and injected into mouse, that does not cause pneumonia. The mouse continued to live. When the heat killed S strains and R strains are mixed and injected into the mouse, that causes pneumonia and mouse dies.
Okay, thank you, Deepti. Biochemical characterization of transforming principle. One more concept again. Veeresh, can you read out the slide, please?
Biochemical characterization of transforming principle. Ashwell, Avery, Collier, Markle, and McLean-Mancarty worked to determine the biochemical nature of the transforming principle of Griffith experiment. They performed biochemical proteins, DNA, and RNA from the heat-killed S strain. They added digestive enzyme of each to see which one could transform live R cells to S cells.
Thank you, Veeresh. So here we have to remember who are the scientists who put forth or who developed the Griffith experiment again. What they used the enzymes. They used the biomolecules enzymes. Okay. Now, Veeresh, can you continue with these 3 points?
Heat killed S strain plus protase live S strain transform R strain to S strain. Heat killed S strain RNS live R strain transformed R strain to S strain. Heat killed S strain DNS live R strain unable to transform R strain to S strain.
Very good. Thank you. So here, why I made you to read this, because like this only they will ask question in the multiple choice options. So that is why I made you to read this. Thank you, Veeresh. Now let us discuss some questions again, guys. Be interactive. Answer quickly. Answer once I finish with the reading question. Okay. The next question is, which of the following is not a part of a nucleotide? ಕೆಳಗಿರುವ ಯಾವುದು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಪಾರ್ಟ್ ಅಲ್ಲ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಆಪ್ಷನ್ ಎ ಫಾಸ್ಫೇಟ್ ಗ್ರೂಪ್, ಆಪ್ಷನ್ ಬಿ ನೈಟ್ರೋಜನಸ್ ಬೇಸ್, ಆಪ್ಷನ್ ಸಿ ಪೆಂಟೋಸ್ ಶುಗರ್, ಆಪ್ಷನ್ ಡಿ ಗ್ವಾನೋಸಿನ್.
Option D, ma'am. Option D, ma'am. Option D, ma'am.
ಗಾಯತ್ರಿ, ಅಕ್ಷಯ್ ಪಾಟೀಲ್, ಕವಿತಾ, ಪಾರ್ವತಿ, ರಾಜೇಶ್ವರಿ, ಪದ್ಮಾ, ನಯನ್, ದೀಪ್ತಿ, ಶಾಂತಿ, ಲಕ್ಷ್ಮಿ, ಸೌಂದರ್ಯ, ವೀರೇಶ್. Okay, Parvati. Prema, most of you voted for option D, few of them voted for option B. So,
Option
here,
B.
Yes, Akshay.
Option B.
Guanosin is the right answer. Good. So, what will be the next step in the process of transcription? DNA into RNA, RNA into what? Question mark. Option A, mRNA, option B, tRNA, option C, rRNA, option D, protein.
Option D, protein, ma'am.
Option
Chandru,
C, rRNA.
Okay, Shanti option A, Saundarya option C, Prema option
C,
A,
ma'am.
option D, Saundarya option C, Deepti option C. RNA ಆದ್ಮೇಲೆ ಏನಾಗುತ್ತೆ? ಆಪ್ಷನ್ ಸಿ, ದ್ಯುತಿ ಆಪ್ಷನ್ ಸಿ, ಫೈನ್. ಓಕೆ, ವೀರೇಶ್ ಆಪ್ಷನ್ ಎ, ವಿದ್ಯಾ ಆಪ್ಷನ್ ಬಿ, ಶ್ವೇತಾ, ಲಕ್ಷ್ಮಿ, ಸಿ ಅಂಡ್ ಡಿ, ಸಾನ್ವಿ ಆಪ್ಷನ್ ಡಿ. The right answer is option C, rRNA, ribosomal RNA. Good. Next, 30th question. What is the length of the DNA double helix if the total number of base pair is 6.6 into 10 raised to 9? Length of DNA double helix if the total number of base pair is 6.6 into 10 raised to 9. 2.2 meter per base pair, 2.5, 2.2, 2.5. Option D, Sanvi option B, Saundarya option B, Amrita
Option B, ma'am. 2.2 meter per base. Option D, ma'am.
ಯಾರು ಸಾಲ್ವ್ ಮಾಡಿಲ್ಲ, ಸಾಲ್ವ್ ಮಾಡಿ ಹೇಳಿ. ಸಿಂಪ್ಲಿ ಡೋಂಟ್ ಆನ್ಸರ್. ರವಿಕುಮಾರ್ ಆಪ್ಷನ್ ಬಿ. Correct taxonomy, you have to multiply it with 0.34, no? Do you know that formula? Length base pair into 0.34. Chaitra option C, Kavita option C. The right answer is, as they asked what is the length of the DNA double helix if the total number of base pair is 6.6 into 10 raised to 9. So the right answer is option A, 2.2 meter per base pair. Sorry, option C. Answer is 2.2 meter. Then the DNA of the E. coli is scattered throughout the cell. Just now we read regarding this in the slide. Or ಏನ್ ಕೊಟ್ಟಿದ್ದಾರೆ E. coli. E. coli, whether E. coli is a prokaryotic or eukaryotic. E. coli prokaryote ಅಥವಾ eukaryote?
Prokaryote.
Prokaryote, ma'am. The DNA of the E. coli is scattered throughout the cell. Sorry. Option A true, option B false.
Option A true, ma'am.
Option A, ma'am.
Option B true, madam.
Option B true ideno?
True.
Veeresh option B, Parvati option B. Is scattered throughout anta kottidare. Just now someone read regarding prokaryotes, right? Guru Sid, Sanvi, most of them voting for option A. Few of them voting for option B. See, just now we read regarding this in the slide, and you guys are not able to answer this. Right. Okay. So as you all said regarding E. coli, as it is a prokaryotic, let us see the structure of DNA in the prokaryotes. You read it, right? Someone read it. I don't remember the name. See, the DNA is not scattered throughout the cell. Prokaryotes do not have definite nucleus. Packaging of DNA in prokaryotes alli second point concentrate ಮಾಡಿ ಅಂತ ಹೇಳಿದೆ. Because we are going to discuss a question on it. I said it, right? So see here, the DNA is not scattered. ಇಲ್ಲಿ ಏನು ಕೊಟ್ಟಿದ್ದಾರೆ question ಅಲ್ಲಿ? The DNA is scattered throughout the cell in the E. coli. Now tell me guys whether this statement is true or false.
False, ma'am.
False.
False, ma'am.
Option A. Few of you voted for option B. So it is a false statement. Good. Okay. Well developed. Okay. So Lohit, can you read out the slide?
Well-developed nucleus is absent in prokaryotes. Genetic material is not organized as chromosomes. Histone proteins are absent. Negatively charged DNA is wrapped around positively charged proteins. Polyamides, like this primitive form of genetic material, is called as nucleoid.
Thank you, Lohit. Okay, the next question goes like this. What are the set of positively charged basic proteins are called? Option A, option B, DNA, option C, RNA, option D, histones. Positively charged basic proteins.
Option D, ma'am.
Option D, ma'am.
Option D, ma'am.
Stones.
Kavita, Amrita, Varsha Kanaki, Saundarya, Kanchana Raj, Deepti, Akshay Patil. Most of you voted for option D. Few of them voted for option B. DNA. Gayatri, Veeresh, option D. Okay. Let us check the correct answer. The right answer is option D. Histones are positively charged because they contain. ಯಾಕೆ positive charge ಇರ್ತವೆ ಅವು? Because they contain a high amount of positively charged amino acids. That we have to remember. ಅದು multiple choice question ಅಲ್ಲಿ ಕೇಳ್ತಾರೆ. ಯಾಕೆ positive charge ಇರಬೇಕು? Because they will be having positively charged amino acids such as lysines and arginines. These amino acids are positively charged because they are basic in nature. Due to this reason, histones are called set of positively charged basic proteins. 33rd question, very easy question. What are the thread-like chain structures present in the nucleus known as? Option A, chromosomes. Option B, chromatid. Option C, chromatin. Option D, chloroplast.
Chromosome.
Option A, ma'am. Option A, ma'am.
Option A, ma'am.
Okay, fine.
Option A, ma'am.
Question ನೀಟ ಗೊತ್ಕೊಳ್ಳಿ.
Option C, ma'am.
Okay, Lohit, option C. Veere, Shanti, Dyuti, Padmayan. At first, the vote went to option A. Then someone messaged as option C. Then I said correct gotkoli question anta. After that, all the answers I got option C. Fine. So option C is the answer, guys. Thread-like chain structure present in the nucleus. Thread-like anno term bandaga, that is option C will be correct. That is chromatin. After certain time, that is during cell division, condensation occurs, where chromatin will be converted into chromosomes. Now let us brush up few concepts again. Okay. Yes, Varsha, connected. Can you read out the slide, please, regarding conclusion of the experiment?
Conclusion of the experiments. Protein of heat-killed S strain is not the genetic material. RNA of heat-killed S strain is not the genetic material. DNA of heat-killed S strain is the genetic material because DNA digested with the DNA is mixed with R strain, unable to transfer R strain to S strain. But all biologists are not convinced.
Okay, the spelling should be corrected. Okay, Varsha, mute yourself and thank you. The genetic material is DNA. Deepti, can you read out the slide?
Protein of it. The genetic material is DNA. DNA is the genetic material is proved by Alfred Hershey and Martha Chase at 1952. They worked on the virus that infects bacteria called bacteriophage. During infection, the bacteriophage first attaches the bacteria cell wall. It inserts its genetic material into the bacterial cell.
Can anyone tell me, this is a bacteriophage? Okay, what is this? The other diagram indicates.
What?
Okay, I'm asking, you can see the diagram of bacteriophage, right? The other diagram is there, which is showing some cyclic pathway. What is this indicates? Which phenomenon is shown here?
Life cycle.
Yeah?
Life cycle, ma'am.
Life cycle? It is a multiplication. Okay, indirectly we can tell it is life cycle. In the
Yes, ma'am.
bacteriophage, we have two cycles, right? Bacteriophage ಹೇಗೆ multiply ಆಗ್ತವೆ? Two process ಇಂದ multiply ಆಗ್ತವೆ. Which are those two processes? Yes, anyone? Did you hear regarding lytic cycle of bacteriophage?
Temperate.
Yeah, tell me, Lohit. You can try it.
Temperate bacteria.
Temporary bacteria ಮತ್ತೆ? Okay.
Lytic or
Lytogenic.
Lytic and you are right. Lysogenic. So these are the two cycles which will show the multiplication of bacteriophage. Bacteriophage ಅಥವಾ virus ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಹೇಗೆ ಮಲ್ಟಿಪ್ಲೈ ಆಗತ್ತೆ ಅನ್ನೋದಕ್ಕೆ two methods ಇದೆ, lytic and lysogenic. Okay, fine. The next one is ಅದು ಬೇರೆ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ಬರುತ್ತೆ. I just reminded you. Chandru, can you read out the slide? Are you here, Chandru?
The viral genetic material become part of the bacterial genome. It manufactures more virus particle using host content. Hershey and Chase worked to discover whether it was protein or DNA from viruses that entered the bacteria. Experiment blenders experiment. They grew some viruses on a medium having radioactive phosphorus, some others on medium having radioactive sulfur. Viruses grow in radioactive phosphorus have radioactive DNA but not radioactive protein because phosphorus present in DNA, not protein. Viruses grow in radioactive sulfur have radioactive protein, not radioactive DNA because sulfur present in protein but not in DNA.
Okay. I will give you 2 minutes. Thank you, Chandru. I will give all of you 2 minutes. Read out the slide because we are going to answer few questions on this slide. Okay. ಈಗಲೂ ಆಗಿರ್ಬೋದು ಆಮೇಲೂ ಆಗಿರ್ಬೋದು. Just read out the slide for 1 minute at least. It is also called Blender's experiment. Then it was put forth by Hershey and Chase. Then they used radioactive phosphorus and radioactive sulfur. Radioactive phosphorus is for DNA because phosphorus is a part of DNA. And by radioactive sulfur, it coded for, it got attached to the protein, not DNA. So this is regarding this slide. Now let us answer a few questions again. Okay. 34th question. How many base pairs are present in a typical nucleosome? Option A 200, option B 100, option C 300, option D 90.
Option
A, ma'am. 200.
Option A, 200 base pairs.
Option A, ma'am.
Kancana Raj, Shamita, Pavitra, Varsha, Dyuti, Mallikarjun, Parvati, Amrita, Kavita, Deepti, Shweta, Guru Siddhappa, Pallavi, Shanti. Okay, most of you voted for option A and the right answer is option A. Next, when the negatively charged DNA combines with the positively charged histone octamer, which of the following is formed? Option A nucleus, option B nucleoid, option C nucleosome, option D nucleosome ಅಂತ ಇದೆ, nucleic acid ಅಂತ ಮಾಡ್ಕೊಳ್ಳಿ. Option D nucleic acid.
Option C, ma'am. Nucleosome.
Option C, ma'am.
Now Lohit read it regarding that answer. ಯಾರು ಓದಿದ್ರು ಅದರ ಬಗ್ಗೆ. Okay. Most of you answered option C, that is nucleosome. Padma, Akash, Varsha, Niresh, Kumar, Gayatri. Option C, nucleosome. One nimisha. It is nucleosome. Okay. 36. How many nucleosomes are present in a mammalian cell? 20 million, 30 million, 40 million, 10 million.
20.
Amruta, option B. Lohit, option B. Prema, option B. Darshan, option B.
Option B, ma'am.
Kanchana, Raj, Padmayan, Megha, Shweta, Likhita, Viresh. Most of you answered option B, that is 30 million. Option B, 30 million is the right answer. Great. 37th question. I'm sorry. Which of the following chromatins are said to be transcriptionally active and inactive respectively? Chromatin ನಲ್ಲಿ ಯಾವುದು transcriptionally active ಇದೆ, ಯಾವುದು transcriptionally inactive ಇದೆ? Option A euchromatin, heterochromatin. Option B euchromatin, prochromatin. Option C prochromatin, euchromatin. Option D heterochromatin and euchromatin.
Heterochromatin.
Option A, ma'am.
Option D.
Option A, ma'am.
Option D. Darshan, option C. Kavita, Megha, option A. Sanvi, Dyuti, Kumar, Pavitra, Lakshmi, Viresh, Basavaraj. Prema, Rajeshwari, Guru Siddha, Asha Patil, Padmayan. Most of you voted for option A. Few of you voted for option B. ಸ್ವಲ್ಪ ಜನ few ಯಾರು C ಅಂತ ಹೇಳಿದ್ದಾರೆ, ಸ್ವಲ್ಪ ಜನ B, some of them C and some of them as CD. Okay. Okay. Darshan and Chaitra, option A. Option A is the right answer. Euchromatin transcriptionally active irutte, whereas heterochromatin will be inactive. 38th question goes like this, guys. What are the additional set of proteins which are required for the packaging of chromatin at the higher level is known as? Option A, histone proteins. Option B, non-histone proteins. Option C, histone chromosomal proteins. Option D, non-histone chromosomal proteins. Additional proteins. So histone proteins jote addition agi proteins irtavalla. So they will be also required for packaging. What those proteins are called as? Nayan, option D. Purvi, option
Option
A.
D, ma'am.
Option D,
Option
ma'am.
A, ma'am.
Non-histone chromosomal
Chandru,
protein.
option A. Pallavi, option D. Shweta, Likita, option D. Amruta, option A. ಯಾರು ಆಪ್ಷನ್ ಎ ಮಾಡಿದಿರೋ ನೀವು ಸರಿಯಾಗಿ ಕ್ವಶನ್ ನ ಅರ್ಥ ಮಾಡ್ಕೊಂಡಿಲ್ಲ. ಅವ್ರು ಏನ್ ಕೇಳಿದ್ದಾರೆ? ಅಡಿಷನಲ್ ಸೆಟ್ ಆಫ್ ಪ್ರೋಟೀನ್ಸ್ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಬೇಸಿಕ್ ಸೆಟ್ ಇಸ್ ಹಿಸ್ಟೋನ್. ಹಿಸ್ಟೋನ್ ಬಿಟ್ಟು ಅಡಿಷನಲ್ ಯಾವ್ದಿದೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಸೊ ದ ರೈಟ್ ಆನ್ಸರ್ ಈಸ್ ಆಪ್ಷನ್ ಡಿ, ದಟ್ ಈಸ್ ನಾನ್ ಹಿಸ್ಟೋನ್ ಕ್ರೋಮೋಸೋಮಲ್ ಪ್ರೋಟೀನ್ಸ್. ನಾನ್ ಹಿಸ್ಟೋನ್ ಕ್ರೋಮೋಸೋಮಲ್ ಪ್ರೋಟೀನ್ಸ್. ಡಿ ಅದು, ಬಿ ಅಲ್ಲ. ನಾನ್ ಹಿಸ್ಟೋನ್ ಕ್ರೋಮೋಸೋಮಲ್ ಪ್ರೋಟೀನ್ಸ್, NHC ಅಂತ ಕರೀತಾರೆ. NHC. The next one, very easy question. What is the typical distance between two base pairs? 0.34 nanometer, 0.32, 0.33, 0.35.
0.35.
35. Okay.
Option A, ma'am.
0.34. Option A, ma'am.
0.34. Option A is the right answer. Option A is the right answer. Fine. The next one is, what is the structure present inside the nucleus known as? ಅವ್ರು ಲೇಬಲ್ ಮಾಡಿದ್ದಾರೆ, ಅದೇನು ಅಂತ. ಕ್ರೋಮೋಸೋಮ್, ಕ್ರೊಮ್ಯಾಟಿಡ್, ರೈಬೋಸೋಮ್, ಲೈಸೋಸೋಮ್.
Chromatid.
It should be not chromatid, ಅದು chromatin ಆಗ್ಬೇಕು. TIN. It is chromatin ಆಗ್ಬೇಕದು. It is not TID. Chromatin. So option B, chromatin ಅಂತ ಅಂದಾಗ what is the answer?
Option B, ma'am.
Option
B, ma'am.
Option B, ma'am.
Okay, Shweta, Megha Chauhan, Amrita, Veeresh, Dyuti, Asha Patil, Gayatri, Guru Siddh. Okay, most of you voted for option B. Option B, chromatin is the right answer. Next, just now we discussed regarding this diagram, guys. Observe it carefully. The DNA helical structure is linked to which type of histone protein in the following diagram? So, ಇದು ಯಾವ histone protein ಅಂತ ಕೇಳಿದ್ದಾರೆ? Option A, H2A histone. Option B, H1 histone. Option C, H2B histone. Option D, H3 histones.
Option B, ma'am.
Option B, ma'am. Shweta, Kanchana Raj, Guru Siddh, Darshan, Megha, Niti, Chaitra, Kumar, Pavitra, Prema. That is option B, H1 histone is the right answer. Great. Then 42nd question, very easy question. Just a minute. Which of the following bacterium causes is responsible for causing pneumonia? Staphylococcus aureus.
Option D, ma'am.
Option D.
Okay, Staphylococcus aureus.
Option D.
Option D. As the name causes pneumonia. Option D is the right answer. Good. Then, when Streptococcus pneumoniae were cultured in a culture plate by Frederick Griffith, which among the following were produced? Yav produce ಆಗ್ತವೆ ಅಲ್ಲಿ? Smooth colonies and rough colonies, shiny colonies and rough colonies, smooth and rigorous colonies, silky colonies and rigorous colonies.
Option A, ma'am.
Option A, ma'am.
There will be certain set of questions in your paper. ಯಾರು ಅಂದ್ರೆ ಅದನ್ನ ಯಾರು ಆನ್ಸರ್ ಮಾಡ್ಬೋದು ಅಂತ. Just certain questions should be like this because ಎಲ್ಲರೂ ಅದನ್ನ ಆನ್ಸರ್ ಮಾಡ್ಬೇಕು. ಆ ರೀತಿ ಸ್ವಲ್ಪ questions ಇರ್ತವೆ. Among those questions, this is one of it. Okay, let us brush up few concepts again. Experiment ಆಯ್ತಲ್ವಾ? Okay, criteria for genetic material. Just a minute. Kanchana Raj, can you read out this?
Yes, ma'am. It should be able to generate its replica or replication. It should be chemically and structurally stable. It should show changes, mutation that require for evolution. It should be able to express itself in the form of Mendelian character. Protein dose does not fulfill the criteria, hence it is.
Just a minute. Yeah.
Protein dose does not fulfill the criteria, hence it is not the genetic material. RNA and DNA fulfill the criteria.
Okay. Read out this slide, please.
DNA has two strands that are complementary. Deoxyribose sugar, OH group at carbon atom. Number 2 in ribose sugar is replaced by only H, which make it less reactive and hence more stable. Presence of thymine in place of uracil also make DNA more stable.
Thank you. Kumar, can you read this replication of DNA? Are you here, Kumar? Okay. Jyoti, can you read the slide? Sanvi,
Yes, ma'am.
can you read out this?
Yes, ma'am. Replication of DNA. It is the process by which DNA produces the exact copies of the original DNA. In eukaryotes, DNA is double-stranded. The two strands are complementary to each other because of their base sequences. Semi-conservative method of DNA replication. It is the most common method of DNA replication. It takes place in the nucleus where the DNA is present. Replication takes place in the S phase of the cell cycle. Deoxyribonucleotides needed for a formation of new DNA strands are present in nucleoplasma.
Okay, thank you. Okay, fine. Can you read out this? Chaitra, can you read out this?
Yes, ma'am. Semi-conservative nature of DNA Matthew and Melson and Franklin Stan. They grow E. coli on 15NH4Cl culture medium. 15N is the heavy isotope of nitrogen. Both strands of DNA have 15N, 15N, 15N. These bacteria are shifted to 14NH4Cl culture medium. DNA extracted subjected to cesium chloride, CSCl, density gradient centrifugations, hybrid intermediate type of DNA 15N, 14N. After next generation, equal amount of light DNA 14N, 14N and hybrid DNA 15N, 14N are formed.
Okay, this is regarding semi-conservative method. Thank you, Chaitra. Yeah, this is important too. This we all discussed. Yesterday, central dogma we discussed transcription agide. Then, fine. So first, now again we will discuss on some questions. Just a minute. Okay, 44th question. The mice infected with the R strain of the bacterium dies, but when infected with the S strain, they do not die. Whether this statement is false or true?
Option A, ma'am, false.
Option
A,
A,
false. No.
Option A, it is false. Very good. Option A is the right answer. Which of the following is the wrong sequential order when the S or the R strain of the bacterium is injected? So ಕೆಳಗಡೆ ಆಪ್ಷನ್ಸ್ ಕೊಟ್ಟಿದ್ದಾರೆ ಯಾವ ಆಪ್ಷನ್ ರಾಂಗ್ ಇದೆ ಅಂತ ಹೇಳಬೇಕು. See the options and let me know the answer.
Option D, ma'am.
Option D. Okay.
Option D, ma'am.
Option D, ma'am.
In chat also we got this same voting, that is option D is the right answer. 46th question. Which of the following is responsible for transforming R strain into S strain? Purified biochemical from S strain, purified biochemical from R strain, purified biochemical from heat killed S strain, purified biochemical from heat killed R strain. ಏನ್ ಕೇಳಿದ್ದಾರೆ ಅವರು ಆರ್ ಸ್ಟ್ರೇನ್ ನ ಎಸ್ ಸ್ಟ್ರೇನ್ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗುವಾಗ ಯಾವ ಫ್ಯಾಕ್ಟರ್ ರೆಸ್ಪಾನ್ಸಿಬಲ್ ಆಗತ್ತೆ, ಯಾವ ಬಯೋಕೆಮಿಕಲ್, ಆ ಬಯೋಕೆಮಿಕಲ್ ಎಲ್ಲಿಂದ ತಗೊಂಡಿರ್ತೀವಿ, ಎಸ್ ಸ್ಟ್ರೇನ್ ಆರ್ ಸ್ಟ್ರೇನ್?
ಆಪ್ಷನ್ ಸಿ ಮ್ಯಾಮ್.
Option C, ma'am. Purified biochemical from heat
Purified
killed
biochemical from killed S.
Option C. Okay. Most of you voted for option C, purified biochemicals from heat killed yes cells is the right answer. 47th question. Once you complete answering, please mute yourself. Otherwise, my voice will echo to me. Okay, which of the following is responsible for the inhibition of transformation in organism? DNAs, RNAs, protease, nuclease. Inhibit ಮಾಡುತ್ತೆ transformation ಅನ್ನ. Chandru, option A. Likita, option A. Likita, option A. Kantaraj, option A. Kumar, option A. Varsha, Amrita, Veeresh, option A. Shanti and Parvati, option A. Asha, Pavitra, Shamita. Great. Lakshmi. Most of you answered it option A. Option A, DNA is the right answer. It inhibits the transformation. Then. Okay. Parvati, can you read out the slide?
Yes, ma'am. Transformation is a phenomenon by which type of cell, when introduced I not another type, is able to give some of its properties or the later in 1944, Avery, McCarthy and MacLeod discovered that protein digesting enzymes, proteases and RNA digesting enzymes, RNAs, did not affect transformation. So the transforming substance was not a protein or RNA digestion. With the DNA did inhibit transformation. They concluded that DNA is a hereditary material.
Okay, thank you. 48. What happens when S strain and the biomolecule lipid are together when testing the transformation with regards to R strain? S strain ಮತ್ತೆ lipid together ಆದಾಗ transformation with regards to R strain ಏನಾಗುತ್ತೆ? ಈಗ ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ, S strain with DNA, S strain ಬರುತ್ತೆ. S strain with RNA, R strain ಬರುತ್ತೆ. S strain with lipid ಏನಾಗುತ್ತೆ? Option A R, option B S, option C R and S, option D dead R strain. Yes. Kumar, option D. Lakshmi, Shweta, D, A. Most of them voting for A. Chandru, A. Veeresh, D. Nayan, B. Let us check the answer. So here only DNAs has the ability to inhibit transformation. The other enzyme like RNAs, lipase do not possess the ability. So the process of transformation is not seen when the biochemical DNAs is with S. Lipase is an enzyme that catalyzes the hydrolysis of lipids and fat. The process of transformation will occur in lipids. As a result, R strain will be produced. So R strain produce agate. Option A is the right answer. Then 49th question. Which of the following is considered to be as incorrect difference between DNA and DNAs? DNA ಮತ್ತೆ RNA ಅಲ್ಲ, DNA ಮತ್ತೆ DNAs ನಡುವೆ ಯಾವ ಡಿಫರೆನ್ಸ್ ರಾಂಗ್ ಇದೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. So here difference, a nucleic acid, DNA is a protein. DNA can be seen in nucleus, DNAs can be seen in cytoplasm. DNA transferring of genetic material. DNAs, it cleaves the DNA molecule. Fourth, it contains only some of the hereditary information. It promotes the DNA. Yes. Kavita, option A. Megha, option D. Gurusit, option A na. Okay. Kumar, option not Kumar. Veeresh, option A. Nayan, option D. Okay. Most of you answered. Most of you tried the answer. Option D is the right answer. It promotes the DNA, like it inhibits the DNA, guys. It inhibits. DNA is in it inhibits the DNA. So option 4, that is only 4, is a wrong difference. Then, what will be the state of the mouse that has been injected with the heat-killed S strain from the Staphylococcus pneumoniae? Alive, dead, on the verge of dying, lives with pneumonia. Yes. Nayan, Kumar, option B.
Option B, ma'am.
I'm dead.
Okay, heat killed. One minute.
Option B, ma'am.
Yeah, ma'am, alive.
Option A is alive. Okay, most of you answered option A, few of you answered as option B. The thing is, it will be alive. 51st question. What will be the outcome when R strain is injected into mice? Mice dies as soon as it is injected. Mice lives in bacteria and dies suddenly. Mice dies after ejecting out the bacteria. Mice lives even after injecting R strain. Be fast, very easy question. Option D. Nayan, Megha, Rohit, option A, Diti, option B, Kumar, option A. R strain ಅಂತ ಕೇಳಿದ್ದಾರೆ.
Option A, ma'am.
Option D. Mice lives even after injecting R strain to the bacterium. Okay, this is about few questions on the molecular basis of inheritance. Now it is time for 5-star bio. Be prepared. And as I am saying, until I complete reading the question, no one is going to answer it. And no one is going to respond through mic. Only you should respond through a message. Fine. Just a minute. Give me 2 minutes of time so that I can present the screen. A few of the question I will be presenting. Few of the question I will be asking. I mean, oral ಆಗಿ ನಾನು ಕೇಳ್ತೀನಿ. Okay, be ready. Until I say you to answer, you should not answer. The first question of 5-star bio goes like this. In one polynucleotide strand of DNA molecule, the ratio of A plus T by G plus C is 0.3. What is the ratio of entire DNA molecule? 0.3, 0.6, C 1.2, D option D is 1. Time starts now. 5, 4, 3, 2, 1. Yes. Answered by Duty. When I completed the 5, 4, 3, 2, 1, the first message I got from Duty, she said, she answered it correctly. Option D, that is 1, is the right answer. Next question of 5-star bio goes like this, guys. Histone proteins are basic and negative, basic and positive, acidic and positive, acidic and negative. 5, 4, 3, 2, 1. Okay. The first message I got when I when the time is up, that is from Chandru. He answered option B. Option B is the right answer, and Chandru is winner for second question. And the third question goes like this. Chemically, RNA is dash 100 dash reactive and dash stable as compared to DNA. Both equally, less more less, more equally. Andre first answer both, second answer equally, first answer less, second answer more, first answer more, second answer less, like this. So your time starts now. 5, 4, 3, 2, 1. So I got the first message from Kanchana Raj. She voted for option B. Option B is the right answer. Now, let me ask you the question orally. Okay. Next one. Which of the following life processes? Hear it carefully. Which of the following life processes are evolved around RNA? Are evolved around RNA? Which of the following life processes are evolved? around RNA? Option A, metabolism. Option A, metabolism. I'm typing option A, metabolism. Option B, translation. I have a book here. Option C, splicing. Option D, all of these. So what is the answer?
Ma'am, option D, all of these.
Metabolism, translation, splicing. The question is, which of the following life processes are evolved around RNA? Metabolism, translation, and splicing. Lohit answered
Option
as
D.
all of these. The first answer is given by all of these, that is option D by Lohit. So the right answer is option D, that is all of these. The last question of today about 5-star bio. Let me ask it. The DNA replication takes place at dash phase of the cell cycle. I repeat, DNA replication takes place at dash phase of cell cycle. Option A, G1. Option B, yes. Option C, G2. Option D, M.
Yes, ma'am. Yes,
Option
please.
B,
Option
ma'am.
B. Yes, yeah. Option B, ma'am.
Okay, Kanchana Raj told us yes phase, option B. The right answer is option B, that is yes phase. So this is regarding 5-star bio of today. Tomorrow we are going to discuss on different topic. I hope the session is clear. Any doubt?
No, ma'am.
No, ma'am.
Did you enjoy the session? Did you enjoy the session?
Yes, ma'am.
Yes, ma'am.
Okay, then let us wind up the session here. Have a nice day. Take care.
Thank you, ma'am.
Thank you.